I'm going to be talking about mitosis and meiosis in this presentation. I am going to go over what they are, their purposes, their process, and I'm going to compare the two. So first of all, what is mitosis? Mitosis is one of the two types of cell division which occurs in most living cells. It involves asexual reproduction and just one round of cell division. And mitosis creates two identical daughter cells with the same number and kind of chromosomes. Mitosis creates diploid cells and are used to make body cells. And meiosis, on the other hand, is the second type of cell division. It involves sexual reproduction and two rounds of division instead of just one. And meiosis also creates four daughter cells that are unique to each other, which they have half the number of chromosomes as the parents. Meiosis creates haploid cells and are used to create gametes. Additionally, crossing over occurs during meiosis. The purposes of mitosis are to grow by dividing and increasing in the number of cells, to heal wounds and regenerate tissue, and to replace damaged or missing cells that might be malfunctioning. And meiosis has a purpose of creating gametes, and by doing so, this allows organisms to continue reproducing and creating life. Now I will talk about the process of mitosis. Mitosis consists of two main steps known as interphase and mitosis itself. And these two main steps are then broken into smaller categories. Interphase precedes the PMAT process. And interphase includes G1, S, and G2. These steps in interphase are like the preparations for the rest of mitosis and are necessary for this process to happen. G1 allows a cell to grow while absorbing more nutrients for the next steps. And then S is when the DNA duplicates itself so that the two resulting cells will have the same copy of DNA. And after interphase is concluded, the PMAT phases start. P stands for prophase, and this is when the nuclear envelope around the DNA starts dissolving while the DNA clumps into chromosomes. Next is M, which stands for metaphase, and in this step the chromosomes line up down the middle of the cell, while the centrosomes travel to opposite poles of the cell, stretching out their spindles to the centromeres of the chromosomes. A stands for anaphase, and in anaphase, the spindles attach to the centromeres and pull the chromatin apart and toward the opposite poles. And lastly, T stands for telophase, and this is when new nuclear envelopes form around the two new DNA clumps. Nucleoli reappear and the cell's ready to divide now. Finally, mitosis ends in cytokinesis, which is when the cell divides into two identical daughter cells. Now I will talk about the process of meiosis. And the process of meiosis is actually similar to mitosis in a couple of ways. One reason is that meiosis also begins with interphase, just like mitosis, and then it goes into its separate phases, often referred to as the PMAT phases. However, unlike mitosis, meiosis goes through two divisions by going through the PMAT phases twice instead of just once. The stages of meiosis that occur in between interphase and cytokinesis are prophase 1, metaphase 1, anaphase 1, telophase 1, and then prophase 2, metaphase 2, anaphase 2, and finally telophase 2. The second round of PMAT in meiosis is just like the PMAT phases in mitosis. However, the first round of PMAT in meiosis is pretty much different because in prophase 1, synapsis connects the homologous chromosomes, and this makes them cross over some of their genes. And then in metaphase 1, the chromosomes line up at the middle but in their homologous pairs. And then in anaphase 1, the chromosomes pull apart to opposite poles of the cell, and in telophase 1 and cytokinesis, the cell splits into two individual daughter cells. And then the second round of PMAT in meiosis, as I said, is pretty much similar to mitosis. However, instead of diploid cells being produced, it's haploid cells. Now, I will talk about what could go wrong during mitosis and meiosis. So for mitosis may actually cause cancer if it loses control. And what cancer really is just mitosis, but with uncontrolled cell division, meaning there are no boundaries or limits to the cells dividing to a great extent. And because of this causes unnatural growths, which can greatly damage the organism's body. And for what could go wrong in meiosis, the most likely error to occur in meiosis is what's called chromosomal non-disjunction, which creates the wrong number of chromosomes in a gamete. And when meiosis does go wrong, it may cause defections and mutations in the gametes, which is pretty dangerous to the organism or the organism that it becomes. And the gametes that are defective and that go through fertilization may end up with a miscarriage and or any genetic disorders. And lastly, I will compare and contrast mitosis and meiosis. They do have their differences. Mitosis involves one round of division, while meiosis involves two. And mitosis creates two daughter cells which are identical, while meiosis creates four daughter cells which are unique to each other. And mitosis creates diploid cells and are used to make body cells, while meiosis makes haploid cells and it's used to create gametes. But mitosis and meiosis also have a few similarities. For one, they start with one diploid cell and replicate DNA before forming cell division. And in both of their processes, the nucleus breaks down and DNA condenses into chromosomes. And in mitosis and meiosis, both of their processes end with cytokinesis. So this was my mitosis meiosis presentation. Thank you.